So we'll go ahead and change gears a little bit and talk about different organelles that are located inside of the plasma membrane now. So now that we're familiar again with that boundary of the cell, we can discuss everything else that's going on. And all these organelles have various functions and contribute to the production, the packaging, and the exportation of certain cellular products. And so you can go through what these mean on your own, but we're going to go into each one of them on various slides now. So starting with the cytoplasm, we kind of dissected this a little bit at the beginning, but the cellular material that's outside the nucleus but inside the plasma membrane is known as the cytoplasm. Within that, the fluid portion is called the cytosol. There are cytoskeletal elements that we talked about before, and these help support the infrastructure of the cell. Think of them as the brick and mortar of the cell that help keep it together. Microtubules are made of tubulin. You can kind of see them dimerizing on each other here. This helps make up cilia and flagella that help move the cell as well as, again, support the cell. Microfilaments are made of actin, which are the smallest cytoskeletal component. This is actually a really large component of muscle cells and important for muscle contraction because they help in motility of that muscle cell. And then intermediate filaments are intermediate between microfilaments and microtubules in size, and they're made of keratin, and they again are helping support the infrastructure of the cells. Centrosomes, these contain pairs of centrioles. The microtubules throughout the cell originate in these centrosomes, and again that helps make up cilia and flagella as well as tracts in the cell that help proteins move throughout. And this will actually end up aiding in cellular division or mitosis, which we'll talk about in lab. Ribosomes, this is where protein synthesis occurs. So ribosomes are composed of a large and small subunit that they assemble together with an mRNA in the cytoplasm out here to help form functional ribosomes. And so that's the rRNA basically. And so once the rRNA is formed, it will use this mRNA to make protein out of the amino acids. So you can kind of see the rRNA here is functioning unit reading the code from the mRNA and making a protein out of that. We'll talk about that a little bit more in detail in lab, but basically think of it as a little protein factory is what a ribosome is. Okay, now the endoplasmic reticulum is a system of channels that are continuous with the nuclear membrane. So you can see the nucleus here into the membrane, and then the endoplasmic reticulum is directly on that, basically. It provides passages throughout the cell that function in like transporting, synthesizing, storing materials. There are two forms, the rough ER and the smooth ER. They perform very different functions, thus they can be present in varying quantities in cells depending on the cell type. The main function of smooth ER, which don't contain ribosomes, is the production of lipids. And steroids are very basically lipids. They're not, but they're made from lipids. And so it synthesizes phospholipids that are part of membranes in cells, as well as steroid hormones from other lipids. This means that cells that produce large amounts of these hormones, such as cells in the ovaries and the testes, have a large amount of smooth ER. The smooth ER also sequesters and regulates calcium, which is incredibly important for neurons when calcium is a trigger for neurotransmitter release. The rough ER has ribosomes attached, so you can kind of see all the little red speckles. And then over here in the micrograph, we can kind of see different stains for ribosomes, the little black dots along the lining of the rough ER. And it's named this because of those little speckles, so it's rough. And those ribosomes, again, synthesize protein. Following the production of the protein by those ribosomes, the protein moves through the passages of the rough ER, where it gets glycosylated. They add sugar to it. The rough ER, I should say, adds sugar to the protein that the ribosome made. And then after the sugar is added, the protein is sent to the Golgi apparatus for further processing. So the Golgi apparatus modifies, packages, and distributes proteins and lipids for secretion or internal use. So you can kind of see that stemming off this rough endoplasmic reticulum, we have a little green globular protein. It gets transported via another vesicle here, into the Golgi apparatus, and then it travels through there, gets modified, packaged, and distributed either out of the cell or packaged up again to be into the cell. And this also, this organelle of the Golgi apparatus also creates lysosomes. Lysosomes are organelles that break down and digest cellular material that is either not needed anymore or is foreign. So maybe the damaged organelle needs to be recycled to use its parts, or a bacterium that was recently phagocytosed needs to be killed. And so you'll see a lysosome come over and digest the material there, and then it'll recycle those parts and use it for the cell. Okay, so it's basically the cell's little digestive system is what a lysosome is. Okay, and then we have the mitochondria, the energy transformer of the cell, the ATP powerhouse, as some say. So this is the site of cellular respiration. Basically, all the ATP that is made, or most of the ATP that is made by the mitochondria. And so there are membranes in the mitochondria that are called cristae, and these are like little infoldings of inner membranes. The proteins that are embedded in here help perform cellular respiration, which converts energy stored in nutrient molecules such as glucose into adenosine triphosphate, the ATP that we discussed in the chemistry lecture. And this provides usable cellular energy to the cell. Cells use ATP a lot, okay? And then the mitochondria are constantly at work because of how much ATP we need. So oxygen molecules are required during cellular respiration, which is why you must constantly breathe it in. One of the organ systems in the body that uses a huge amount of ATP is the muscular system, because ATP is required to sustain muscle contraction. As a result, muscle cells are packed full of mitochondria. Nerve cells also need a lot of ATP so that they can run their sodium-potassium pumps that we discussed for active transport. Therefore, an individual neuron will be loaded with over 1,000 mitochondria. It's kind of crazy, actually. On the other hand, a bone cell, which is not nearly as metabolically active, might only have a couple hundred mitochondria. And lastly, the famous nucleus, which is the cell's largest and most prominent organelle, and is generally considered the control center since it stores the genetic information for manufacturing proteins. Some cells in the body contain multiple cells, like multinucleated cells. These are like muscle cells. Other cells, like red blood cells, contain no nuclei at all. They eject their nuclei when they're young to make space for all the hemoglobin they're going to carry, and they don't end up lasting long. And so for that reason, the body has to produce new red blood cells constantly. The nuclear envelope has small pores in it so that RNA can enter and leave the nucleus to make copies of DNA, and so that ribosomes can get synthesized, or so that ribosomes that are synthesized in the nucleus can leave the nucleus and then go to the cytoplasm where they can manufacture proteins for us. DNA is in the form of chromatin when it is in an uncoiled mass, and when it is preparing for mitosis, it condenses to form chromosomes. So when it's not going to be dividing, it's in the form of chromatin and kind of like a soupy spaghetti mixture. And then when it needs to be mitotically dividing, when it needs to be prepared for that division, it condenses those into easily movable structures called chromosomes. And so after this nucleus now, we don't have any other organelles to discuss, and that actually concludes our discussion on the cellular level of organization.